Hello everyone, this is Akshay over here and welcome to my another video. So in this video we will be mainly looking into biomolecules. So let's get started. The biomolecules are basically the carbon compounds which are mainly observed in the living organisms. What we are mainly referring here is the living tissues of the organisms. So we call them as biomolecules. So can we visualize the biomolecules or can we look into the biomolecules in reality? So the answer is actually yes. So what we can do over here is that you can take a tissue and you can grind it with a chemical called as the trichloroacetic acid and you get thick slurry, and this slurry can be divided into two important components. The first one is the acid insoluble pool and another one is the acid soluble pool. So in these two pools you can see the biomolecules which are mainly present. So not only the tissues are made up of the carbon compounds or carbon derived compounds, but at the same time you can also find inorganic constituents in the living organism or the tissues of the living organism. So how can we actually determine or analyze the inorganic constituents of the cell? So that can be done by drying of the tissue, and after the drying of the tissue you have to incernate the tissue, basically the burning of tissue, what I'm referring over here. So this results in the formation of ash. So the burnt remain of the tissue is referred as the ash, and that is what remains finally. When you analyze the contents of the ash, you can see there are inorganic contents. So this table over here mainly shows the different components and their formula, the inorganic components and their respective formula. So you can see that sodium is present, potassium is present, calcium is present, magnesium, water, and compounds such as sodium chloride and calcium carbonate and so on. So these are all the things that you mainly find in the living tissues of the organisms. So we can look into one such important biomolecule over here, that is the amino acid. So amino acid is having both group, that is acidic group and basic group. So the acidic group and also you can see the amino group or also the basic group. So this diagram over here is mainly showing the structure of an amino acid over here, the general formula or general structure of amino acid. So this carbon here mainly shows the alpha carbon. So towards the left side of the alpha carbon, you can see over here is the amino group or the basic group, and towards the right side of this particular alpha carbon, you can see the carboxyl group or also the acidic group. And you can even see that the alpha carbon, on one end of the alpha carbon you can see there's a hydrogen, and on the other end of the alpha carbon you can see there is a side chain which we refer it as the R group. So in nature you find 20 amino acids, and each and every amino acid is having its own unique R group, and this R group is important and is responsible for giving unique properties to the amino acids. So one important aspect that is associated with the amino acid is the Zwitter-ion concept. So Zwitter-ion is basically a German term which means hermaphrodite. Why do we refer an amino acid as hermaphrodite? Because an amino acid which is carrying both the positive and negative charge, such ion is called as Zwitter-ion. Okay, so you can see over here the diagram B over here, you can see that the one end of the amino acid is carrying the positive charge and another end of the amino acid is carrying the negative charge. So as the same amino acid is carrying both the positive and negative charge, hence the net charge of this amino acid is going to be 0, right? So you can see that at a different physiological pH in the living organism, amino acid can be in the Zwitter ion or it can be in one particular charge state. So the A diagram, what you can see over here, you can see that it's containing only one charge, that is plus charge. Hence the charge of this amino acid is going to be +1. But the C diagram, you can see over here that the amino acid charge is minus. Hence the charge of this amino acid is going to be -1. So now why does this amino acid can be in +1 charge state or -1 charge state, or also why the amino acid can be in net 0 charge? So this is completely a very complicated topic to discuss and which is beyond the scope of this tutorial. We can dedicate this discussion to a completely new video. But anyway, we have to remember over here that the amino acid which is containing both the charges, that is the positive and negative charge, such amino acid is called as the Zwitter-ion. And also remember the pH at which this amino acid is observed as a Zwitter-ion or the pH at which the amino acid carries the both positive and negative charge, that pH is referred as the isoelectric point, or also called as the pi. So this is what we have to remember over here. So moving on, we are having the 20 amino acids over here, and the classification of 20 amino acids are as follows. The first group is the non-polar, one more is the polar, and another one is the electrically charged amino acid. So what do we mean by electrically charged amino acid? The R group of these amino acids are having the positive charge or negative charge, depending upon the type of the amino acid. So the polar ones are the ones that can be dissolved in water, and the non-polar are the ones which are not dissolving in the water. So we can look into one such amino acid, that is the glycine. You can see that the glycine, it's referred as one of the most simplest amino acid, and the reason is that you can see over here that the R group of the glycine is simply hydrogen. Hence, glycine is referred as one of the simplest amino acids. And moving on, we are having one more amino acid, that is the alanine. And alanine, you can see that the side chain of the alanine is simply the methyl group, CH3 group. And in the case of polar amino acid, one example we can discuss is the serine, whose side chain is hydroxymethyl, that is CH2OH. So we can look into one electrically charged amino acid, that is the acidic amino acid. An example for this is the glutamic acid. So the glutamic acid is a negatively charged amino acid because the R group mainly contains a negative charge. So also one important point regarding the glutamic acid, that it is a neurotransmitter in the nervous system, and also the same glutamic acid is also the precursor for another neurotransmitter called as the GABA, that is gamma aminobutyric acid, which is an inhibitory neurotransmitter. Anyway, not associated for biomolecule. Still an important point, we can consider it over here. So glutamic acid is a negatively charged amino acid, or also acidic amino acid. And finally, one more amino acid we can look at here is the lysine, which is a basic amino acid or positively charged amino acid, because the side chain or the R group mainly contains positively charged. Hence, it is a basic or positively charged amino acid. So here we can look into three important amino acids, that is the tyrosine, tryptophan, and phenylalanine. And why they are important? Because they are aromatic amino acids. They mainly contain the ring over here, the ring structure. Hence, that is what it makes them the aromatic in nature. So this is all about the amino acids. So let us move on with the lipids. So lipids are water insoluble, as we all know, and one type of lipid that we can see over here is the fatty acids. And fatty acids are basically the carboxyl group that actually are attached to the R group. And the R group can be either methyl or ethyl. So some examples for fatty acids are palmitic acid, which is mainly made up of 16 carbons. So 16 carbon also counts the carboxyl group. And one more fatty acid is the arachidonic acid, which is mainly containing the 20 carbons, including the carboxyl group, carboxyl carbon. So the fatty acids may be either saturated or unsaturated ones. For example, palmitic acid over here is actually a saturated fatty acid, while the arachidonic acid is polyunsaturated fatty acid. So another molecule that we have to consider over here is the glycerol. So the fatty acids are esterified with the glycerols, and when fatty acid is actually esterified with the glycerol, you get monoglyceride, diglyceride, and triglycerides. Okay, and some of the lipid molecules also contain the phosphate groups. So here, a moment ago, I told about diglyceride, triglyceride, and monoglyceride. If you attach a phosphate molecule to the diglycerides, you get an interesting type of lipid called as the phospholipids. So now, phospholipids are very much important for the construction of the plasma membrane. Okay, and one such phospholipid which is mainly found in the plasma membrane is the lecithin, or another name for lecithin is also the phosphatidylcholine, or also called as phosphatidylcholine, right? So this is about mainly the glycerol. So you can also see that in the cells there are some organic compounds which are also made up of the heterocyclic rings. Okay, so nitrogenous bases are examples for the organic compounds which mainly contain the heterocyclic rings. So we all know the nitrogenous bases are basically of two categories, that is the purines and the pyrimidines. So among the purines and pyrimidines, you can see adenine, guanine, thymine, cytosine, and uracil, which are the nitrogenous bases. So these nitrogenous bases are very much important component in the nucleic acids, that is the RNA and the DNA. So this is about mainly the nitrogenous bases. Moving on, we can look into another part of the biomolecule, that is the metabolites. So you find two types of metabolites in the living organisms, that is the primary metabolite and one more is the secondary metabolite. The primary metabolite mainly includes the sugars, amino acids, lipids, and the nucleotides. So why do we categorize them as the primary metabolite, all these as primary metabolite? Because inside the cell, these metabolites, these biomolecules are constantly used for one or the other metabolic pathway or one or the other cellular metabolic activity. So as they are used inside the cell for one or the other activity, hence they are actively taking part in the cellular processes. Hence we refer them as primary metabolites. While contrary to this, we are having is the secondary metabolites, whose function is not well defined. We don't know what they actually do. So some examples over here we are having for the secondary metabolites are the alkaloids, flavonoids, rubber, essential oils, and the antibiotics. So this table over here shows some of the examples for secondary metabolite. So in the case of pigment, which is a secondary metabolite, examples mainly include the carotenoids, anthocyanins. And in the case of alkaloids, we are having the morphine and codeine. So morphine we all know for its anti-pain activity. So next we are having is the terpenoids, the monoterpenes and diterpenes. And essential oils, we are having the lemongrass oil, and we are having toxins, that is the abrin, and one more we are having is the ricin. Next we are having is the lectins, that is the concanavalin A as an example for lectin. Next one we are having is the some important therapeutic drugs called as the vinblastine and curcumin. And finally, we are also observing some polymeric substances as also secondary metabolites, such as the gum, rubber, and cellulose. So these are some of the representatives of the secondary metabolites. This is all about the introduction part of the biomolecules. So we will stop our discussion over here only, and shortly we will be meeting with another video where I will be mainly discussing about the biomacromolecules. So we will meet in the next video. Till then, goodbye.